And thanks, Marcy, for that introduction. Thank you for inviting me here today, and thank you all for coming to this Tools and Tech seminar. I've been excited for several months now to present a couple tools I've been working on in Lydia Fredalino's lab. I spend most of my time really thinking about how we get the most out of our sequencing data and how to help biologists get the most out of their sequencing data and designing tools that will be really helpful and intuitive to biologists to use and to interpret the results that we get out of them. And I think we're getting close with these two tools, Enricherator and ShapeME. So what these tools really aim to help biologists do is to understand protein-DNA interactions at a couple different levels. But I want to point out how important protein-DNA interactions are to our understanding of a wide array of biological processes and phenomena. So here I've just got this toy example where we've got some unnamed pink protein floating around in space and genomic DNA here. And this protein has a specific binding site indicated in red. And whether this protein binds that site or not will have an impact on whatever biological process the protein's involved in. So let's assume it's involved in regulating gene expression. It could promote gene expression by recruiting RNA polymerase. It could repress gene expression by blocking RNA polymerase recruitment or blocking its progression along the DNA. If it's involved in any number of DNA metabolic pathways, such as DNA replication, it could promote or inhibit DNA replication. It could engage in DNA repair. The point is, all these processes you see here and many more that you could think up and imagine are underpinned by protein-DNA interaction. And these protein-DNA interactions will, in large part, define the behavior of many of these biological processes. So throughout biology, protein-DNA interactions are really essential for us to understand whatever process we're studying. All right. So understanding the interactions requires us knowing where they occur in genome space, usually, to really understand them holistically. We need to know where they occur. So here I've got another toy example where we've got kind of simplified, perfect-looking ChIP-seq data. If you're unfamiliar with ChIP-seq, the intuition you need to know about it is that it identifies for us where a protein of interest binds DNA. So for this protein of interest, it binds here and here. All right, so we know where this protein binds. That's great. Now, what if we want to know how the protein binds the DNA? One method we can use, and we often do use as biologists, is to look for what we call motifs that are predictive or even perhaps define that protein's interaction with the DNA. And I've drawn that as just an orange box here, representing some sequence element in the DNA that encourages the protein to bind that site specifically. So knowing where the protein binds and how it binds are really important to knowing what this protein is doing and what biological process is affecting and how. All right, so it's not enough just to have ChIP-seq data, right? That doesn't give you an inference on where the protein is binding. We need to process the data. And current tools for processing these types of data fall a little bit short, in my mind, of really giving us the inferential value we seek from processed ChIP-seq data. And I'll go through some intuition with you as to how that happens and how this tool Enricherator solves the problems we get with the current methods of analyzing ChIP-seq data. So Enricherator, what it basically does is infers enrichment of the biological signal of interest. So here we're talking about protein-DNA interaction relative to some baseline expectation. And we get that baseline expectation just by sequencing the entire genome, right? So that's kind of the baseline we expect by random chance. We just get from the genome sequencing data, and from ChIP-seq data, we can then use those two pieces of information to calculate the enrichment of the signal of interest. Gaps in current methods, the major gaps to me that exist are that very often with most tools that are available and most ways that we handle these data, we handle the count data that sequencing data are as if they are continuous in the math we use to kind of infer enrichments of interest. And you'll see in this talk how that can be extremely problematic. And the second major limitation in current methods is the vast majority of them completely ignore the fact that sequencing data has a structure to it. When we sequence DNA, we're sequencing fragments that have a defined size. And that structure can actually tell us a lot about the coverage we expect to see in our sequencing data, and we can use that to our advantage when we fit models to the data to infer enrichments of interest. The second tool I'll tell you about is called ShapeME. That stands for Shape-Based Motif Elicitation. It's designed to be a complement to current sequence-based motif inference tools. It's not designed to replace them, but instead of looking only at sequence of DNA for motifs, it looks at local shapes in the DNA for motifs. And you'll see the GitHub repos for both of these are on this slide if you're interested, and you should be. Take a look at those repos. All right, so I want to give you the upshot right off the bat as to how Enricherator can be useful compared to the old way of handling these data. So I'm abstracting a lot away from you, but this is a real world example that actually inspired me, I'd say forced me to write Enricherator in the first place. Where what we have, let's just call it ChIP-seq data. It's not, but that's the method I think most of us are familiar with. So let's call this ChIP-seq data. On the y-axis for both of these, you have basically inferred log fold enrichments of this protein binding that we're interested in. And it's the same genome position we're looking at in both of these plots. So it's exactly the same data that I processed. And the top, I used kind of the old standard way. And the bottom, I used Enricherator to process the data. All right, so right away up on the top, I think what you'll appreciate is we see a bunch of these dramatically, unrealistically negative discrete spikes in the inferred log scale enrichment. And we know these are unrealistic just based on thermodynamics, basically, right? Like there's not zero enrichment of the actual signal of interest. There's something there. Like if I took a coin and flipped it 3 times and got heads each time, none of you in this room should say, that coin will never flip a tails, right? You know that's unrealistic. And yet the math does not, right? The math that we're all using to analyze these data does not recognize that property of the count data that we are using. But Enricherator does. And what it basically just does is fits a generalized linear model to sequencing count data. And it does this using an inference engine called variational inference. You may have heard of this called variational Bayes. It's a way to fit a model fairly quickly with Bayesian techniques. And you get samples from what's called an approximate posterior distribution out of that. And so a couple things that are nice about Enricherator, in addition to providing more realistic enrichment estimates than you see with the old way, you get these point estimates of your enrichment represented by the solid lines. And you get these 90% credible intervals basically for free from the samples from the approximate posterior distribution. And so we get really intuitive estimates of our confidence in our enrichments, along with more accurate estimates of our enrichments. All right, so that's kind of the intuition behind what Enricherator does on these data. And by the way, those data I was showing you, they were not bad quality data. They're actually abnormally high quality. And yet the old way causes these kind of issues with these dramatically negative, unrealistic spikes in the inferred enrichment values. All right, so I've given you kind of the intuition for what Enricherator can do with our data. And I want to now build in some intuition for how the old way actually gives us these unrealistic values that it does. So here's a separate toy example. Here's a given genome. The DNA is just this line. This red protein has 2 specific binding sites in this genome. So there are 2 locations where it is highly enriched. So the actual biological enrichment of interest that we are trying to estimate with ChIP-seq would be represented in this, fake line that I just drew by hand, where effectively there's 0 enrichment at the non-specific sites everywhere in the genome, except these 2 specific sites where we have a high enrichment. So that's the actual signal we are trying to estimate when we do ChIP-seq. All right, what the actual data may look like then is when we do our whole genome sequencing, which we often call input DNA. When we take our input DNA counts, we see this is our baseline expectation for just sequencing coverage writ large across the genome if there were no enrichment at all. When we do our ChIP-seq, we do see the reads pile up at those specific binding site. This is probably the best case scenario where you have a decent antibody, the experiment works well. And then we see, some other sequencing reads kind of work their way in. They could come from a number of processes. DNA might stick to the tubes that you're using in the lab. That would be coming from some noise process. They could actually come from true non-specific binding of the protein of interest, right? So we don't know exactly where these reads come from, but they come. All right, and now we see with this, and this would be what I would consider a fairly high quality ChIP-seq data set. And what we see is there are regions of the genome where the coverage drops to 0, right? So especially in these regions of the genome, we get real problems when we start to assume that the sequencing data are continuous values, because they are absolutely not. They never are, but they're definitely not at these low coverage regions of the genome where the counts are 0, 1, 2. And so you can see that in the math here that, when we just calculate these enrichments in the old way, we take some normalized measure of the ChIP-seq coverage divided by some normalized measure of the baseline expectation input coverage. And because, again, we're pretending these are continuous values, and we shouldn't be pretending that, we have a little workaround to avoid dividing by 0 or having 0 in the numerator, where we just add a small pseudo count. Like, well, we know this is unrealistic math for the actual data generating process, but we're going to just add the pseudo count to try to pretend that it's realistic. We're going to make it look more realistic than it is. All right, so we do that. And what results is that these regions where we get 0 coverage in our ChIP-seq data set, you can see when we take the log scaled enrichment using continuous math the way we did, we absolutely just get these strong negative spikes. So again, this comes from a couple of things. We're using math that's not really appropriate to the problem. And also, we are not sharing information between local nearby positions of the genome. So by show of hands, I know several of you will raise your hands here, but by show of hands, who here has worked with like sequencing count data like ChIP-seq before in your own research? Raise your hands high so I can pick on one of you. You in the back. If I told you that the sequencing coverage at position I is 100, what would you guess the sequencing coverage at position I plus 1 is?
It should be very similar.
It should be very similar. It should be about 100, plus or minus a couple, right? We automatically know this. If you have any experience at all working with sequencing data like these, you know that there is a high degree of information shared between nearby genome positions. That if you know the coverage at one spot, you automatically have a very good guess of what the coverage is at the nearby spot. But the current math does not appreciate this. It just can't. It's not written into the assumptions of the model. Okay. So Enricherator actually it solves both of these problems where it shares local information in a principled way based on the structure of sequencing data that we know exists. It's in our mental model. It should be in our mathematical model, right? And it also handles count data appropriately. So it gives us these more realistic estimates. Okay, so let's get under the hood a little bit now that you have the intuition behind what Enricherator does and what it's designed to do. I already mentioned it uses variational Bayes to fit a model to the data. And again, this is just nice to handle these data in a Bayesian way so that we can get samples from an approximate posterior to give us kind of intuitive, credible intervals on our point estimates. It's also nice that we can use Bayesian stats here because we can apply a shrinkage prior on our enrichment estimates. If you're not familiar with what that means, don't worry too much about it. You've probably heard of correcting for multiple hypothesis testing, though. What shrinkage priors do is basically that in a Bayesian way, right? So that we don't have to then, correct our enrichment estimates post hoc. The math can do it explicitly with a prior that basically assumes there's very little enrichment unless the data has a strong reason to pull our posterior away from that shrinkage prior. All right. It uses a negative binomial likelihood so that we properly handle our count data. And here's the math. So basically at every genome position, for every genotype we look at, for a given sample type, whether it's input DNA or ChIP-seq DNA, and for each strand, if you have strand-specific counts, some methods allow that. For something like ChIP-seq, you usually don't. So, you would just have non-stranded data. And for each sample ID, for every sequencing library we sequenced, we say that the observed counts are drawn from a negative binomial distribution centered around the expected counts with some overdispersion phi. And we fit a separate overdispersion for each sample type here, because we know a priori that ChIP-seq data should be more overdispersed than input sequencing data. We just know that to be true based on experience. And every time I fit this model to any data, that's actually borne out in the posteriors. So that's appropriate to have that separate overdispersion term globally for the different types of sequencing data we're working with. All right, so now our expectation here with its log length, since we're working with count data, basically is expressed in this way, where we have a certain input DNA abundance that we fit. So the model kind of learns the input DNA abundance. And then based on whether we have ChIP-seq data or not, we either consider or not, respectively, beta here. So we learn beta based on the actual ChIP-seq enrichment over what we get in our input DNA. And then here CJ is just an expression for, it's an offset term to account for library size. It does the library size normalization for us. So if you sequence one library to 2 million reads and 1 to 1 million, you expect the one in my left hand to give you half the counts on average at each genome position compared to the one in my right hand. And this term accounts for that explicitly in the model here. Okay, so you don't have to normalize your data basically before putting it into Enricherator. It's built into the likelihood. Okay. And then again, I'm going to reiterate, and this is really, I think, the truly novel thing about Enricherator is it uses sequencing fragment length information to locally pool information in our enrichment estimates. And this is how it does it. So I already mentioned this alpha term. So this now is an alpha, it's a vector alpha, it's a dense vector. For every genome position, this alpha has an estimate of the input DNA abundance. And then if we look on the right side, we have alpha prime. This prime is not meant to indicate a derivative. We're not taking a derivative of anything. It's just meant to say, this is a different alpha than this alpha, but they're related. So alpha prime is a sparse vector. So basically, instead of looking at every genome position, what we're actually going to do is fit a value for every maybe 5th, more likely every 50th genome position, depending on the actual fragment size in your sequencing libraries. So if the fragment sizes are long, we're going to infer alpha primes wider apart than if our fragment sizes are short. And then we build a weights matrix to basically take the dot product of what I'll call a pooling matrix. And I'll explain with a cartoon in a slide here what this really looks like. But we get this pooling matrix. So the dot product between that matrix and the sparse vector alpha prime gives us our dense vector alpha of actual genome scale resolution enrichment estimates, or in this case, input DNA abundance estimates. And then depending on whether, again, we're using input values or whole genome sequencing counts for alpha, we get a weekly informative prior on those values. But if they're enrichment estimates, betas, we have a shrinkage prior that we apply at this point before taking this dot product to get back to our genome resolution enrichment estimates. So in a picture, here's kind of what this looks like, where again, the sparse vector alpha prime is up here and it's represented by these circles. So each value that we learn in sparse vector alpha prime is represented by a color up here. And then the pooling kernels in the weights matrix W alpha are represented as the different color exponential kernels, these double exponential kernels. Right, and what I intend to convey here is that, again, we're looking at input DNA in this case, but for the enrichments, for the ChIP-seq, you can imagine, we'll have different fragment sizes. And so these distances will be different. But for the input DNA, the distance between each inferred value in sparse vector alpha prime is the input fragment length over 2, just half the fragment length. And then the way I construct these pooling kernels is such that the mean width of them is equal to the average fragment size in the sequencing libraries that you're working with. So it's a very principled way to use the fragment sizes in our sequencing libraries, the structure of the data, to then take and mix the actual fitted values in the sparse vector back to our genome resolution in basically what would be a deconvolution of this sparse vector. Looks like we have a question.
Why are kernels sharp?
Because it's a cartoon that I drew.
Okay, so are they actually like Gaussian kernels? Or?
Oh, so no, they They are exponential, so they are sharp.
Okay,
And the reason I do that is because I think, there's a certain probability with which, if you have a certain fragment, you don't know where the protein exactly was bound along that fragment, but it was probably in the middle. So, we could split hairs over whether it should be Gaussian, or Hamming, or what have you, Laplacian, whatever. But I've chosen exponential, because I think it just makes sense to me to consider there being an exponential decay from the middle of some signal. Are there any other questions at this point? Okay. All right, so now I just want to point out one other nice thing about Enricherator using Bayesian inference here. That makes it really useful, I think, to biologists who oftentimes are interested in how a signal changes in a condition-dependent way. So these data are from a collaboration that we have ongoing with Athmaja and Chris from Michigan State University, where we're looking at this molecule. Chris Waters' lab, they're experts in cyclic DiGMP, which is a signaling molecule that bacteria have. It doesn't matter what it does for this example, they have it and it does stuff. All right, so what we've done is we've done ChIP-seq on RNA polymerase to look at where transcription is happening along the genome of this bacterium. So there's just one genome position that we're particularly interested in. And you can see that these are the Enricherator results for this position of the genome, either with cyclic DiGMP or without. So I think we can all agree that with a high degree of confidence, we can say that when cyclic DiGMP is high, RNA polymerase is more abundant at this location that's probably the promoter of this gene. It's a bit more abundant in the middle of the gene than without. But wouldn't it be nice if instead of just looking or forcing someone to look back and forth between these, we could actually calculate that contrast that we're actually interested in? What we're actually interested in is the effect of cyclic DiGMP, not just one condition and another, right? We're looking at comparing them. So with these Bayesian samples from an approximate posterior, we can actually do that. For every sample, we can calculate the difference between these conditions, or the contrast between these conditions. What that then gives us is samples of the difference from which we can get our point estimate of that effect of interest and our 90% credible interval of the effect of interest. And again, we're basically getting this for free, this intuitive look at the actual factor that the biologist wants to know about for free because we're using Bayesian inference here. So there's a lot, I think, that Enricherator has going for it. Of course, I think that I wrote it, but I think objectively, this tool has a lot going for it. One caveat, though, is, again, because we're using Bayesian inference, it is a little slower than just dividing a bunch of numbers by a bunch of other numbers, right? So I've been ragging on that way of doing things, but there's a reason we've all been doing it, and it's not because we're stupid. It's because we don't have time to wait for our computer to fit a model for a week, right? So on a bacterial-sized genome with several samples, a reasonably sized data set, Enricherator takes several hours to fit, which is actually not bad, right? But if you're talking about a human genome, Enricherator is not going to be useful for you, frankly. We'd have to figure out some way to speed things up significantly. For like an S. cerevisiae genome, a yeast genome, I think it could be useful for you. So keep that in mind as you look into Enricherator and decide whether to use it. Also, as I work toward publishing this, I'm working on including a noise term, because right now all the data affects the input abundance and the enrichment estimates. But we know that some reads come from these noise processes like sticking to the tube that we're using. And so the addition of a noise term should really help the predictive performance of this model. And in addition to that, people are starting to use more and more often these spike-in normalization methods rather than just normalizing to library size. And we're working on incorporating that into the Enricherator model as well. Are there any questions about Enricherator at this point before we go on to ShapeME? Go ahead.
Yeah, so there's an example you asked about earlier, like where we have 100 counts at I, and then you probably have close to 100 counts at I at this one location, right?
Yeah.
With this algorithm, are you worried about losing spikes that are biologically relevant?
Yeah, that's a good question. And in case the owl didn't pick it up, I'll repeat it. Basically, the question was, are we concerned about loss of resolution, like genome space resolution, with this kind of pooling, local pooling of data? And every time I fit this model, I actually look at the posterior samples. We get posterior predictive samples of the sequencing counts to see how well they match the actual data. And Enricherator is just doing a very good job. And so I think, yes, I think absolutely we are going to lose some informative spikes that actually mean something biologically. But if those spikes are informative and are in your data and are well supported by your data, you have short enough sequencing fragment sizes to actually, your smoothing should just work out to capture it. And if those spikes are in your data and your actual resolution doesn't support their existence, you don't want to infer that those spikes are biologically meaningful. You want to smooth it out. And Enricherator will do that. So there's, as with anything, there's a give and take and benefits and drawbacks to what we're doing here. But I think overall, you're going to get a better view of what your data actually support the existence of by pooling the information that Enricherator does. Mitch.
So for your prior distribution for like the alpha like vector is, you said you had like a variance of 4, right? Is that like hard coded into like all the models, or is that like supported by literature, or is there any like hyperparameter tuning for that?
Yeah, that's a good question. So Mitch asked about basically the choice of prior on alpha, essentially DNA abundance relative to library size, which is what that prior really represents. So that choice, it is hard coded. And honestly, I didn't put that much thought into the width of that prior. And I really should, because for input DNA, we really don't expect massive changes, the type of wide changes that prior explicitly denotes we're expecting. So I probably should narrow that down to something that's better supported by experience with these types of data. So for like a bacterial genome, we'd expect 2 to 4-fold change across the genome if they're replicating their genome, and this prior far exceeds that kind of difference. So, okay, let's go on to ShapeME then. I mentioned that it uses local DNA shapes to infer motifs that are predictive of protein-DNA interaction. So most of you will be familiar with this kind of view of a motif. This is a sequence motif. They're defined by position-specific weight matrices. This is just an example of a motif I downloaded from a database called the Jasper database, where for this transcription factor, AFT2, what the logo here means, and we call these sequence logos, or some people call them memes, I think, but sequence logos. What this means is, if you see a piece of DNA that has A or G here, followed by C, A, C, 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 you really expect AFT2 to bind that piece of DNA. And that's just what this means. It's a very intuitive way to look at how sequence is predictive of protein-DNA interaction. Some issues with these are that they assume that each position of the DNA is independent from every other position of the DNA. And we just know that's not true biophysically, right? If we change a base pair at one position, you'll get local changes in the shape of the DNA up to 2 or so nucleotides away from that single changed base pair. So it's just not true that these positions are independent of each other. And of course, sequence motifs, they only capture, or sequence itself only captures a fraction of the actual information content in a piece of DNA. So our hypothesis has been that local shapes of the DNA may outperform sequence motifs for certain proteins. And that's actually a pretty conservative hypothesis. I think actually most people in this room will appreciate that it's probably a foregone conclusion that is going to be borne out in the data. So I'll just give you the upshot right off the bat. It is true, it is borne out in the data that ShapeME, using shape motifs, can perform better at predicting protein-DNA interactions for many proteins than sequence motifs can. So let me explain what you're seeing here. The y-axis here is just a given human transcription factor. I went to the ENCODE database and downloaded ChIP-seq data for each of these factors and looked at, okay, where are the peaks in the human genome for each of these? And let me fit ShapeME to the data to try to infer sequence motifs, which are in the red, or shape motifs, which are in green, or in the blue, ShapeME actually allows you to run mixed models with both sequence and shape. And then in the final step of ShapeME, it'll kind of do a lasso regression to do model selection and pick from the shape and sequence motifs that were identified, which are the most informative, right? So it might report to you one sequence and one shape motif, or all shapes or all sequences when you run it in this blue both mode. Then the filled circles are each of the 5 folds for 5-fold cross validation I did, and the open circle represent just running the performance of a model trained and evaluated on the entire data set. All right. And then on the x-axis, I've got basically each model's performance for each protein, where performance is measured as the area under the precision recall curve. So 1 would mean perfect performance. And for these data sets, the dashed line indicates random performance, or just garbage, basically. All right. So what I really want to point out, and again, here's the upshot of what ShapeME can provide to biologists. For this specific transcription factor, POU5F1, we see that shape performs really well, with AUPRs of 0.75, which is great. Sequence motifs also do pretty well. AUPRs are above 0.5. That's great. When we run ShapeME in the both mode, we get really good performance, but it's only as good as the shape performance was in the first place. And if you actually dig down and look at the motifs, ShapeME is returning to us the shape motifs. So what this indicates is that for certain proteins like POU5F1, SP4, YY1, any information that sequence was able to give us was probably just shared information that sequence has with shapes. And if we look more directly at the shapes, we get a more informative view of protein DNA interaction. All right, so now you kind of see, again, the upshot of what ShapeME can do. Let's take a step back. We'll talk about what these shapes are and how we're using them to infer motifs. All right, so by local shapes, here's basically what I mean. DNA has a minor and a major groove. So we can look at the minor groove width. For B-form DNA, on average, it's about a little over 4 angstroms. Propeller twist is when the pairs, the bases in a base pair, twist oppositely to each other along their base pairing axis. Roll is when they twist together, the same rotational direction along their base pairing axis. And helical twist, of course, is for every base pair you move along the helical axis of the DNA, how many degrees does the helix rotate? Importantly, these shape values at each position in the DNA can actually just be calculated, basically deterministically, from the sequence. All right, and if you're interested in how that works, that's not what this talk is about. I would point you to these citations, okay? So for now, for the purpose of this talk, trust me, this works. All right. And I wanted to provide another bit of intuition for why we think looking at the shapes will provide a dramatically different view of the DNA than just looking at sequences. So here I took and I looked at every single 5-mer that exists for DNA, and I calculated in the lower triangle the edit distance or the Hamming distance between each paired up 5-mer. And these distances are just scaled so they kind of are able to be projected on this color scale evenly between each other. But if every position of the DNA is different, that's an edit distance of 5. If every position is the same, that's an edit distance of 0. Whereas the upper triangle, I've taken all these 5-mers, converted them to shapes, and calculated the pairwise Manhattan distances between all of these, where the Manhattan distance is just the absolute value of the difference between each shape value at each position, all summed up to give us that one Manhattan distance. So what I think you'll all appreciate is that the distance landscape for shapes just looks dramatically different than the distance landscape for sequences. All right. Now, for some proteins, sequence is clearly what matters. Like if you ever use a restriction enzymes in your work, you know that like that thing recognizes sequence, right? So this is the way that some proteins appear to be looking at the DNA, so to speak, if you'll forgive the anthropomorphism here of a protein. And this is the way that some other proteins are looking at the DNA, more on the shape level. All right, so again, ShapeME is more of a complement to sequence-based motif inference, not a replacement for it. So here's the basics of the ShapeME workflow. I'm going to start by just showing you what the input sequences look like and the scores that you can give to the ShapeME algorithm. And then we'll go through a few more of the details that I think are kind of important to understand. So first of all, we'll just look at the sequences. And of course, in real life, you would want thousands of input sequences, maybe hundreds. You could probably, if you have, a really good, clean data set, maybe hundreds could give you some good information. For here, we just have 3 for the sake of instruction. So for each of these sequences that I would input to ShapeMe in a FASTA file, I could have 3 scores, or well, one side. For each sequence, there's 1 score, right? So the scores could be binary. The most intuitive example here is like, is this sequence from a peak or is it not, right? That's a binary input score that ShapeMe could make use of. The scores can be categorical. Maybe you have different types of peaks. You can think of other examples. The scores can be continuous, but ShapeMe will actually discretize these and bin them to make them categorical scores on the back end when it works with these data. So it's pretty flexible is the point here. There are different types of scores that you can give it to associate the scores with the sequences. All right, so once ShapeMe has your sequences, it'll convert each sequence to a shape. And this is the way that I represent shapes these days, where for this 7-mer, each position has one of 5 shape parameters shown on this plot. And you'll see on the y-axis, these have been z-scored already. And so like EP stands for electrostatic potential of the minor groove. I know it's technically not a shape, but it's useful for this. So bear with that. Helical twist here, minor groove width, propeller twist, and roll. And I know the resolution isn't great on this, but we have the colors so you can kind of tease apart which icon belongs to which shape parameter. So for each position, we have each of those 5 shape parameters. And each one has a value on the y-axis, of course. So that's how we convert these sequences to what I'm calling shape seeds of a specific width. So once we have all those shape seeds, we can take every seed in the input data and run it down and compare it to every sequence in your input data, which is a fairly computationally intensive process, but it works very well. So for every sequence, we can say, okay, does the Manhattan distance between the seed and the reference sequence fall below some threshold to call it a match? So if we look over here on the right, you'll see for this top sequence, we're pretending, frankly, that this seed matched on just one of the 2 strands. And so for that, we give it a match score of 0, 1. For the next sequence, it matched on each strand. So that gets a 1, 1. And for the next one, it didn't match anywhere. So that's 0, 0. We can express that then as just a hits vector. And now this becomes very useful. And this is really the heart of ShapeMe, if you will, where we can take this hits vector of just categories and compare it to the scores vector that you gave to ShapeMe. Right? That's your ground truth. And we do this comparison using a metric called adjusted mutual information, where if knowing this hits vector tells us everything, gives us perfect predictive power for predicting these scores in the scores vector, the adjusted mutual information score is 1. If there's no information shared between them, the score is 0. So I want to build out a little more intuition about what mutual information really is. The classic example of mutual information is, today it's very sunny. So you would all, I think, assume that it's not raining, right? So sunniness and not raining have a very high degree of mutual information. Just knowing that it's sunny on a given day lets you predict right away with high accuracy whether it's raining on that day. So that's a classic intuitive example of what mutual information does for us. I also want to point out that you could say, well, why not use like a correlation coefficient or something, right? So mutual information doesn't require a linear relationship, right? And it can handle data much more flexibly. It can handle categorical data, right? So it's just a very flexible way to basically assess the similarity between 2 vectors. Okay. So in the real world, when you actually prepare your sequences to feed into ShapeMe, this is kind of what it might look like. We don't actually know where the motifs are a priori, right? This would just be a black line of genome space. We know we had chip peaks along this DNA. So we gather up the sequences under those peaks represented here, and we gather up a bunch of randomly chosen sequences that are outside of peaks as our negative set. So then we get our peak vector. So these are the scores, again, that you input. So you got the sequences here and the scores. Those are the inputs to ShapeME. And then this final vector is kind of that hits vector between a seed or a motif and each sequence that we input. So here you can see, if the motif was actually under these peaks 100% of the time, and rarely was it outside of the peaks, there's a high degree of mutual information here. And we say that's an informative motif. Right? And so here's the map for mutual information. If you really looked at it for a while, you would notice that it doesn't just measure like the similarity between these vectors per se. It's more like how often if they change, do they change together? Right? And that's really the information that it's giving you. All right. So we put our data into ShapeME. It gathered up all these seeds of shapes. It got these hits vectors. It calculated the adjusted mutual information between every seed and our input data. All right, that's great. Now we have usually tens of thousands to hundreds of thousands of seeds that we need to filter through and decide which were actually informative. And of those that are informative, which are distinctly informative and which are redundant with seeds that we already identified. All right. And I'm not going to go through the details. I'm just going to tell you we use something called conditional mutual information for that, which just addresses the question of, whereas mutual information says, if I know one vector, what do I automatically know about another? Conditional mutual information says, okay, if I know one vector and another, now what does this other one tell me about the first, right? It's conditioned on one. It just says, does this next thing provide additional information to the model that the first did not? And that's how we prune the redundant seeds. Then we go through an optimization routine to tune every seed's shape values and the weights that are applied to those values when we do a Manhattan distance calculation. So I'm just going to show kind of the intuitive representation here. Hopefully it's large enough to see where, if we take this, again, just toy example seed, you can see that after optimization, some of these values have been nudged up or down. But not only that, you'll see that the opacity of these rectangles has changed. And what I'm encoding there with the opacity is the weight associated with this value of this shape at this position in our Manhattan distance calculation. So for example, here propeller twist at position 1 has a weight of 0, basically, so it's transparent. So the optimizer basically figured out that the value of propeller twist here just doesn't matter. So we're going to drop it out of the distance calculation entirely, not even consider it. So now this is what I consider a proper motif. It's not a seed anymore, it's grown up, and now it's a motif. So after that final optimization of all the seeds, what we often see is that some seeds will converge to actually be the same motif. We can further prune the redundant one from that using another round of filtering using conditional information, followed by lasso regression for our final model selection. And then ShapeME gives a nice HTML report to the final user so they can kind of open that up in a web page. And the goal here is to get a web app running so that people can just submit their sequences online and get the result and download their results so that the biologists can use this very easily. All right. So I just want to show you now that you kind of have seen the ins and outs, so to speak, of how ShapeME works. I'm going to show you that it works. First of all, I generated a number of synthetic data sets where I just put known sequences at known places, and ShapeME gets those back, it finds them. The most interesting test data set to me that I created was one where I took 50 of the most diverse sequences I could find that basically all had the same shape. Because that's really the crux of ShapeME. That's the thesis here, right? That we're identifying shape motifs no matter what sequence they came from. So in this test data set, when I run ShapeME in shape and sequence mode, it finds one motif, and it's this shape motif. Right? So it does exactly what we expect on that type of data set. And just for reference, when I run it in sequence only mode, so in this case, it's actually just a wrapper for Stream, by the way. Stream is a currently existing sequence motif finding tool. I'm not rewriting Stream, right? I'm just using ShapeME as a wrapper for that. So when running sequence only mode, it does actually find some fairly informative sequence motifs. It's just kind of interesting that the optimization routine I use to identify these sequences, I maximize sequence entropy and minimize shape distance. It just happened to kind of converge on GC-rich DNA. Just kind of interesting, maybe from a biophysics angle, I don't know. Worth thinking about some. So here's actually the quantification of the performance of these various modes of running ShapeME on that test data set. Where again, I'm showing you area under the precision recall curve on the x-axis. Random performance is indicated by this line. And here the circle is the mean of the cross-validated performance, and the bar is plus minus the standard deviation of the 5-fold cross-validated performance. So we see shape only performs the best. Sequence still performs fine, but sequence and shape performs as well as shape does, which again, we expect that given that sequence and shape is actually just using shape information for this data set. I already showed you the kind of human transcription factor data when I kind of showed, here's what this does. We know that for some proteins, shape really matters. For some, sequence really matters. And for some, there's a combination. Again, this is really complementary to current techniques. And then we have these 3 bacterial proteins that have been really hard to identify good sequence motifs for. And ShapeME does an okay job finding shape motifs for them, which is kind of interesting. Especially our lab is really interested in this HNS protein, which coats bacterial DNA. And we're finding a decent shape motif for that in our ChIP-seq data sets as well. So overall, I think ShapeME is going to be really useful for biologists. You should try out Enricherator and ShapeME if you have the appropriate data to use them with. Here are their links. Go check them out. We have a singularity container for ShapeME that hopefully will make it very easy to use with the instructions that I've supplied on GitHub. But if you try these out and you find the instructions confusing or the container difficult to use, please reach out and let me know. I'm very interested in hearing that feedback. And thank you everyone for your attention. Thanks to everyone in the Fredalino Lab, of course, but especially Lydia, Mike, one of our former graduate students, and Rebecca for all their work on the various aspects of what I presented today. Are there any questions at this time? And yeah, any raucous applause? Frank Shin has a question or comment.
Statement is to impose the relationship based which could also be alternatively achieved by storing the drawing probability of new retention.
Yeah. Yep. Yeah, absolutely. So what Changshin is pointing out is that basically calculating these shapes from the sequence is really just a convolution of the various k-mers up to like 4 to 5 mers that you could just express it as sequence k-mers in your input data, basically, if I'm understanding your point correctly. So others have done this. So people have, and it won't surprise you to hear that people have incorporated 2, 3, even 4 mer design matrices, if you will, into their sequence motif inference tools. And they perform pretty well. The problem is that your parameter space explodes exponentially when you do that. And so you're really getting, your model becomes more and more sparse. Whereas by directly looking at the shapes, we don't really explode the parameter space as significantly. Okay. Great. Well, thank you. Mitch, did you ever?
I kind of have an out there question, and you could also be a question. Like, is there a way to sort of kind of do the reverse of what ShapeME does in the sense of like, say if you have like a protein structure, whether or not it can like recognize or will interact with a specific like shape motif of like certain DNA? Kind of working reverse.
I think the entropy space is just way too large there. I don't think so. Not currently. But you're right, Changshin would be the expert. But based on the look on his face, I'm going to say no. Okay. Well, thanks again, everyone.